Rachel. Hi, everybody. It's really nice to see you. So I'm Karin Holmfeldt, and I'm at the Linnaeus University in Kalmar. And I'm going to talk a bit about my work on phages. So phages is viruses that infect bacteria, and that you see here. I hope you see my pointer. Here is a electron microscopy of one of my phages, and here it is where it's growing on the bacteria, forming these beautiful holes that are called plaques. So starting with, I mean, I don't know how much you know about viruses or phages, but we're going to give you some background. And viruses are the most abundant biological entity on the planet. There are about 10 billion phages for every star in the universe. And their combined length is about 10 billion light years if you would put them right after each other. So it's pretty lot of viruses out there. And virus ecologists have a tendency to do fun calculations. So I have a really good colleague who calculated that while there is 10 to the 7 viruses per milliliter of seawater, there's about 10 to the minus 19 great white sharks per milliliter of seawater. So quite that many more viruses. Also, if you take all the carbon content of all viruses in the ocean, that would be equivalent to the carbon content of 75 million blue whales. Now, I don't really know how many blue whales there are out there, but I'm sure there are not 75 million. So viruses, I mean, the viruses that I am interested in is the viruses that infect microorganisms. And these have a huge impact of the viral community. And I really like these cartoons who was published by Maya Breitbart, that's showing the impact of viruses on the microbial community. And one thing is that the viruses lies about 20 to 40% of the bacterial community every day. That's quite a lot. And by doing that, they affect the microbial food webs and also like the general marine food webs. So here we have a schematics of a marine food web, starting with phytoplankton, going to zooplankton, fish, and then bigger organisms like orcas. And if you know about the microbial loop, you will see that the phytoplankton release dissolved organic matter that then gets eaten by bacteria and then transferred up to the food chain. However, now viruses come in and interfere. So instead of moving up this food chain, viruses lyse these bacteria or a large portion of the bacteria and recycle the organic matter so that the bacteria will basically eat their dead buddies, and thereby they're increasing the bacterial growth. Viruses are really interesting because they can do more things than just killing their hosts. And one thing is that they really, like a wizard, take control of their bacterial hosts. And they do that by both expressing their own metabolic genes and also by regulating the bacterial metabolic pathways. They're basically telling the host, stop what you're doing, don't care that you're a bacteria, now you should do everything that would please me, so you can make more viruses. And the most well-studied system for this is most likely viruses that infect single-cell cyanobacteria like Synechococcus and Prochlorococcus. And this system is quite interesting because when the viruses infect the cyanobacterial cell, the cyanobacteria is kind of trying to commit suicide because it stopped the production of the PSBA gene or the PSBA protein. And that's the protein whose uses sunlight in the photosynthesis pathway. Now, when it stopped producing the PSBA protein, basically the cell don't get any energy. And it's kind of thought like, okay, bacteria don't think, I know that. But the idea is that, okay, this cell is doomed. It is going to die anyway. So it could die a bit faster. And then it doesn't produce any viruses. So its buddies won't get infected. Now, this virus is so smart. So it has its own PSBA gene with it, actually, and kickstarts the photosynthesis again. It also affects some of the other pathways in the host, and thereby producing a lot of viral progeny that then gets released and kill these other bacteria cells. Now, the interesting fact here is that by doing this, it also acts, on our biochemical cycling, for example, oxygen production. So approximately 5% of the oxygen that you guys breathe has actually been produced by a virus. Okay, a virus in a host, but still by the virus. Viruses are also really important for gene transfer. Bacteria are not that good at having sex. And that's what sex is about, transferring genes, mixing your genetic material. But viruses can instead be the guys who's transferring this genetic material, coming here like a drug dealer, come on, take some genes. And by doing that, it can give the bacteria, for example, genes for toxin production, substrate utilization, and even antibiotic resistance. One of the most famous examples here are, for example, Vibrio cholera. That's the bacterium causing cholera. But it's actually not the bacteria who's making Vibrio cholera dangerous for humans. It's a virus that's integrated in the bacterial genome that is making Vibrio cholera dangerous to humans, because that's where the toxin gene is. Now, as you see, the impact of viruses on the microbial community is huge. But to be able to have an impact, you actually need to be able to infect your host, right? And even though there are some, benefits for the host to get infected, the bacteria is putting up a pretty good fight to not get infected. One of the first things you have to do as a phage to actually gain entry is to absorb to your host. And you do that through receptors. So for example, this blue phage would absorb on this blue receptor. This was a review by Labrie in 2010, which is really good to read if you're in this with phage host interaction and infection. So now this phage can attach to this blue receptor. Then the bacteria can change the receptor to a purple receptor, and then the phage can't infect. Then the phage can change its tail fibers too to be able to recognize this new receptor. And then the host can block this by proteins or other things. So you see, it's a constant arms race between these phages and hosts. But say that you have been able to attach and now you're adding your DNA into your host. Then there are other things that the bacteria also do. And one is this restriction modification system. Meaning, so here you have the phage, the DNA is getting into the host, you have this infection, but then the host might have restriction enzymes, who's basically cutting up the DNA. So what does the phage do then? Well, then it's adding, for example, methyl groups that would prevent this restriction enzyme cutting. I'm not sure how much you guys have used, but if you done basic like molecular work, then you might have run into this with restriction enzymes, because that's been used in the labs for years. And actually, this was even awarded a Nobel Prize in 1978. Just this simple way of bacteria preventing virus infection. There are also other systems that can be used. And I just want to mention this so you know about it, though I won't talk that much about it later, but it's the CRISPR-Cas systems that got the Nobel Prize last year, actually. And this is also coming actually from just how bacteria prevent infection by phages. And here the bacteria is taking part of the virus genome and adding it here between these spacer, and then they use it to recognize, and once again, they're cutting up the bacterial DNA. So this is also something that's used in molecular ecology, just coming from, phage-host interaction. So it's really important to know what these phages and bacteria are doing. Now, my interest lies in viruses and bacteria from marine environment or aquatic environments, and mostly from the Baltic Sea. And the system I'm going to talk about this is my collection of Celophaga baltica phages and hosts. So I have 19 bacteria. They are highly similar on the 16S rRNA gene. They are all yellow, very pretty. And for these bacteria, they were all collected around here in the southern part of Sweden. We have isolated 45 different phages. This turned out to be a really interesting collection of phages. as these are really, really diverse. So we have all these, this is a map of where you see how many genes these viruses share with each other. And as you see, even though they infect the same host, a lot of them share very, very few genes with each other. So there's really high diversity. They represent actually, we have genome sequences of 12 different genera of phages, and actually one more, this one we have just got sequenced. It's turned out to be a really pain in the ass to sequence. So we're going to be working on that. These are also interesting because they come in all these different flavors of shapes. In virus ecology or virus taxonomy, I would say, before we had genome sequences, people looked a lot at the tails of these phages to place them in families. We know that they have no genetic relatedness within these different families, but we are still using a lot of these things. What's interesting here is these non-tailed phages that actually have single-stranded DNA. All these other are double-stranded DNA phages. So it's a really nice collection to be able to do a lot of experiments with. And one thing we have been looking is as host range. Remember, as I said, that my bacteria were highly similar on the 16S rRNA gene. If you would just do 16S sequence, you would say this is the same bacteria. So this is a cross test where we have all these bacterial host strains up here, and here are all my phages. And everywhere where it's black, the phage have been able to infect the bacteria. However, with different efficiency of infection. So if it's dark black, means that all viruses we added were able to infect the host. If it's gray, just a small portion of these viruses were able to infect the host. And as you see, for a virus, all these bacteria are really different. You know, we have some viruses that can infect nearly all of them, and some are really, really highly specific and can only basically infect the host that were used for isolation. So how can this pattern be created? And that's what I kind of want to talk to you about and show you some of the data we have. And the first one we should look into is, why do we have this different grayscale? Why can this virus infect some of the hosts really well and some not? And I'm going to use one of the systems that is a short-tailed phage. It's called 4:1. So we have the original host of isolation, where basically all the viruses that we add can infect a bacterium. Then we had another host where only about 0.1% of the viruses can infect this bacterium if we like mix the bacteria and viruses. Now, the interesting is that we took viruses that had infected here and isolated them, and then we reinfected both these hosts again. And now we could see that all of a sudden, 100% could infect the original host and 100% alternative host. So what was this? Had this become like kind of a new kind of virus? We were not really sure. So we sequenced both, and it turned out they were both identical on the DNA sequence. There were no differences. And really fun is that if we now isolated this phage from here and reinfected it again, like doing a loop here, we were back to the first thing that, well, 100% could infect here and only 0.1 there. And then we could go around and around and around, and the same pattern came all the time. And what we think, but we're not really sure, because unfortunately we didn't really find any really good answer to this, is that this is due to this, you know, with the restriction enzymes. So probably these two bacteria are providing the viral genomes with different methylation or glycosylation or something like that, preventing the restriction enzymes to be cut. So this bacterium has a different restriction enzyme than this bacterium. So this phage, most of them get cut up by this bacterium, but as soon as one has replicated through it, it's getting the right modification and can then infect really great again. Now, our problem is that we don't really find any differences in known methylation and glycosylation. So this is something we're still going to be looking at. But by doing this, our bacterial hosts are really creating this mosaic pattern where we have these different grayscales. Now the question is, okay, so how can we have, I mean, this was one of the reasons why we get the grayscales. How can we get this, that it can sometimes infect a host and sometimes it cannot? And one of the reasons why we're going to look one of these reasons we're going to look into now. That's an experiment we did where we used a chemostat. Now it's important to know what a chemostat is. So when you do experiments, you could do it in a bottle, for example. You have your media in a bottle, you add your bacteria and your phage, and the bacteria will grow. And while it's growing, it's using up nutrients and it's producing rest products, and then it will increase in abundance. So the composition of everything in this bottle will change, but you have the same liquid. If you have a chemostat, you're basically adding new media all the time and you're taking away old media. In that way, you can have a stable composition. You can keep the nutrient level at the same state all the time. You're taking away waste products. And you can keep your bacterial culture at the same abundance all the time. And then just change one factor. For example, how the bacteria are changed by adding different phages. So we added 2 of these phages from our collection, one called SM and one called ST. And we're going to look at these results now. So the overall bacterial abundance was pretty stable during this whole experiment, which was run over 700 hours, except here in the beginning where you have a big dip in the bacterial abundance. And this dip was basically caused by this phage. So this phage was added at a low abundance, but they fast replicated in their host, increasing in abundance, causing a crash in the bacterial community. Now, when we look into these bacteria more specifically, so you see this green line, it's really low here in the beginning, but then it's higher. It's the abundance of bacteria that are resistant to this particular phage. And you see after this crash, all these bacteria are basically resistant to this virus. So we have caused resistance in bacteria. 2 viruses, remember, this is either virus and behaves totally different from the first one. So instead, by increasing really fast in abundance, this one is a really slow growing one. So it's increasing quite slow in abundance. And also the resistance to this one is increasing slowly. And at the end, we only have basically bacteria that are resistant to both these viruses. So we isolated then bacteria from different time points. Here early, before we had any resistant, during the middle where we have resistance to one of the phage, and late where we had resistance to both of the phage. And then we looked at, because we had many more viruses that could infect the original bacteria. So we looked at how did this resistance change also for other viruses. So we see here in the beginning, this is the original strain. It could be infected by all these phages. And here is the first phage, and it's also still could be infected by all these different phages. But as soon as we get this first resistant bunch, we get resistant only to this phage and 2 more phages that basically are identical to it. It's the same kind of viral species. However, here in the end, we get resistant to all these different phages. So it's like we're closing one little door, but here we're closing a big door that's an entry route for all these different phages. So now it's the question, what is the entry door? And it turns out that a lot of the time, these entry doors of viral infection are linked to things that bacteria use to get nutrients inside them. So we looked for, can these bacteria use different carbon sources, for example? So this is the carbon sources that the original bacteria strain can use, that every black block is a different carbon source. And if it's black, they can use it. If it's white, they can't. And this first phage or this first bacteria that's not resistant could use all these carbon sources. However, when we get this first strain, we can see, or these first strains that are resistant just to one type of phage, they lose the ability to basically take up and degrade all these different carbon sources. The interesting now is that these that were double resistant or multi-resistant still have the ability to use all these carbon sources. So clearly, these bacteria has evolved first, but these bacteria did not evolve from the first one. They were kind of evolved from something that was lying down here, and then they got resistant by closing a totally other door. They don't have this lost inability to utilize carbon sources. However, there's a really interesting fact that this bacteria has lost, because these bacteria can degrade agar. And why would that be important? How do we see that? Well, basically, if we grow these bacteria on the agar plate, we would see like a halo around it. It's like a dark patch, and that's showing that they degrade agar. And these bacteria has totally lost that ability. And why would that be important, like in the ecosystem, for example? Well, these bacteria turn out to grow on macroalgae, like fucus, different fucus plants. So the ability to degrade agar can be a really important nutrient source, you know. So there is a really big cost in being this resistant. And we can see here how actually the viral pressure from outside can cause these changes in resistance if the virus have the ability to infect a host or not. So we have both this, that we're closing these substrate channels, and we have this ability of methylation and restriction enzyme. And that is causing this shift we see in the phage host interaction patterns. And with that, I think my time is more or less up, and I just want to acknowledge the people who has been supporting me with all this work. My PhD supervisor Lasse Rienemann and his PhD supervisor Matthias Middelbo, who's been doing, I mean, both, I worked with the cultivation with them and also this the chemostat experiment. And then Matthew Sullivan, who was my postdoc supervisor at the University of Arizona, and then Yaron Pinhasi at the Linnaeus University, who's been supporting me when I came back here. And I think I'm done.